I'd like to thank you for coming and welcome you to the September 19th meeting of GLIMPRINT and the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics. We're very pleased that Cameron Griffiths from the University of Virginia has agreed to talk to us today. As always, I need to remind people that this meeting is live streamed on YouTube and an edited version of it is made available publicly. So please be aware of that. As always, Tom Helikar and myself are available for your comments, suggestions, and ideas. Jim Sluka and Lorenzo are the other members of the executive. We're all here to try to make things useful for people. We have our usual set of communication channels. They work better when they when you use them. I was going to say when they're used. We have the handle MSM Viral. We have a Slack channel, which I don't think is used very much. We have the IMAG MSM wiki page. We have a LinkedIn page. We have our growing archive of YouTube videos. And we have the GLIMPRINT page, which is being worked on by Jim Sluka and others. We have, again, that MSM Viral Twitter handle. Please help us publicize the talks and also the videos after they're posted so that people will be aware of this work. Please do help us build the glimprint.org. We have a variety of executable models online. If you would like to submit models that you have developed, please do so. If you have papers that you would like publicized, if you have jobs that you would like to advertise, if you have meetings that you would like to advertise, please do send us those announcements and we will make sure that they get put up. This community is extremely dynamic and active, and we realize that you have a lot of places you can publicize things, but we're happy to do our bit to try to help you get the attention that you deserve. As Jim mentions here in his slide, we can post job announcements on the GLIMPRINT site. There's no charge for that. You can submit them on the GLIMPRINT site, or you can email them to jimsluka@iu.edu, and we'll be glad to do whatever we can to build this community. Reinhard Labenbacher is still hiring in the area of computational biology, so please do contact him if you're interested. It's a great program. I think people know him very well. He was one of the co-founders of this organization, and please do talk to him about these positions. Jason Shoemaker is looking for people to work on influenza infection. Please contact Jason. He's spoken in this group before if you're interested in those positions. There is a new call for the POSE, Open Source Ecosystems Grant from NSF. I happen to have one of those. They're very useful. They support community development, which is not so easy to fund, and software development, which is not so easy to fund. Please do take a look at that. The due date is January 14th for this round. That's a once-a-year opportunity. There is going to be a workshop in virus dynamics in Bordeaux next year. If you're interested, please take a look. That, again, may be of interest to a fairly large number of people in our community. Of course, coming up in 2 weeks is the big IMAG MSM meeting. I'm sure I'll be seeing a lot of people there. That's really a team building meeting, trying to develop teams to compete for the new RFAs from NIH and other agencies in biomedical digital twins. The Army will be there, the NSF will be there, DARPA will be there, DETRA will be there. And so I think almost all of us, since this started out life as an IMAG MSM working group, most of those people who show up here will be part of that at one point or another. And the big lead organizer is Gary Ann, who of course has been very influential in this group. There's also going to be a smaller artificial intelligence and precision medicine meeting which can be done online. I think that the other one can also be joined at least partially online. If you're interested in diabetes and chronic diseases and you'd like to be part of that, please consider participating. As always, these slides are available if I'm going too fast so that you can't catch the links. There's a nosy on systems modeling, infection, and immunity. So really, our world is pretty busy at the moment, I would say, looking at all of these opportunities. There's going to be this U54 related opportunity. The deadline is pretty soon for the first round, but that's going to be a regular, periodic, normal due date solicitation. Again, if you have publication you'd like to have listed either on the IMAG site, the GLIMPRINT site, or both, please do send that to jsluka@iu.edu. We'll make sure it gets notified there. Coming up in 2 weeks, we have Dagmar Eber from ETH Zurich talking about her work, a talk from the Karolinska. If somebody would like to suggest somebody, you've heard me say the same rubric again and again, suggest yourself, suggest your graduate students, suggest your postdocs, suggest the people you'd like to hear. Please let us know. We have that October 31st date. Oh, I'm being corrected. Lorenzo says actually Vodovots is going to be talking on the 31st. So that is filled. I'm sorry. Vodovots is replacing Dagmar Eber on the 3rd of October, but we still have an open slot on October 31st. And we're not going to meet on Thanksgiving. So thank you, Lorenzo. You could break in and correct me too. Okay. Please do send us your suggestions. Lorenzo's been working diligently to fill up our roster. Be creative. Can be experimentalists, clinicians, industry people, people doing software infrastructure, anything related to this very broad topic of multi-scale modeling, digital twins, medicine. Are there any other announcements that anybody in the room wants to make? So tomorrow morning at 11 a.m., we will have a steering committee meeting. If you think you should be invited and you haven't received an invitation, let me know, and we'll make sure that you receive the link for that. We have been asked to submit a proposal for a series of 3 conferences on immune digital twins by the Army Research Office. We're very grateful to them for their interest. And so the topic of the steering committee meeting tomorrow will be organizing submissions for those conferences, actually more workshops than conferences. So again, there's a lot of interest in this area and its development, and we want to push that ahead. So please do come and volunteer your time to work on those proposals to the ARO. Okay, I don't want to take any more time from our speaker. We're very glad that Cameron is here today to talk about viral heart infection. I'm not sure that we've actually had any talks about, we've had a lot of talks on respiratory diseases and some other viral diseases, but I don't think we've had a specific talk on heart infection. And so I really appreciate having somebody come and talk to us about that. And I will turn the screen share over to you.
Thank you for the introduction. My name is Cameron Griffiths, and I'm a postdoc at the University of Virginia in Kevin Jane's lab. So I think you might have heard a little bit about viral heart infections from Kevin Jane's before, but it's certainly been a while, and I don't think you've seen any of the stuff that I'm going to present today. So my talk will be about systems biology and viral heart infection. And so first I wanted to say, what does systems biology usually mean to me? So there's a lot of different definitions, and there's going to be a specific definition for the first part of the talk, and it's going to shift a little bit for the second part of the talk. So the first part is a data science-based systems biology, where I like to start with large data sets, use bioinformatics to generate hypotheses that you can test using wet lab experiments, find molecular insights, and do in-depth statistical analysis to figure out the knowledge gap, and with that knowledge gap, generate a data set to solve the knowledge gap or find a public data set to solve the knowledge gap. So viral heart infections. Viral heart infections are actually a very complicated problem. There's diverse patient and viral characteristics. I mean, every person is different, which is kind of the idea behind precision medicine and digital quick twins. And there's actually a lot of different viruses that can infect the heart. There are viruses that infect the heart tissue directly, the cardiomyocytes. These are cardiotropic viruses. There are viruses that infect blood vessels within the heart, vasculotropic viruses. There's also viruses that prefer immune cells and cause heart inflammation by infecting immune cells in the heart, and viruses that don't infect the heart directly but cause indirect cardiotoxic effects and result in heart inflammation. There's also a range of patient responses when someone encounters a heart virus. Generally, it starts with an acute infection, whether or not you, it can be asymptomatic, but if you do have symptoms, it frequently feels like another viral infection, kind of weakness and fatigue. And your heart can spontaneously recover. It can resolve the infection, or the infection can simmer in a chronic state, kind of a low boil, for years at a time. And this can either spontaneously recover or lead to heart failure. And viral heart infections are a very important cause of a certain kind of heart failure called dilated cardiomyopathy. And the final thing is viral heart infections are actually quite difficult to study. As I mentioned, they're often asymptomatic or like other viral infections, so they're frequently undiagnosed. And the only way to diagnose them is with a heart biopsy. And I'm actually not lining up to, get a heart biopsy just to know if I have a virus in my heart. So kind of there's a big need for non-invasive methods to figure out if you have viral heart infection. And specifically, the usual way that you tell if you have heart inflammation is an MRI, but that can't tell you whether a virus or something else is causing it. And so there's a large unmet need to figure out what human infections really look like so we can assess animal models or cell culture models of viral infection. And so this leads me to the systems biology approach to viral heart infection. Unfortunately, there are very few human data sets with diagnosed viral heart infection. However, there are large data sets with human heart transcriptomes. So we ask the question, do these hearts harbor undiagnosed viral infections? I managed to find 3 data sets comprising about 1,000 hearts from healthy and dilated cardiomyopathy. I merged them together and removed some of the ones with lower quality reads, resulting in 979 individual heart transcriptomes. I then looked in the unmapped reads, so the reads that don't map to the human genome, and looked for known cardiopathogenic viruses. And actually, in about 1 in 5 hearts, I found evidence of a cardiоpathogenic virus infection, which is shockingly high. It was way more than I anticipated. And I found viruses from all 4 types, so vasculotropic, lymphotropic, cardiotoxic, and cardiotropic. Interesting to note, almost all of the viruses I found are actually DNA viruses. So the fact that I'm picking up RNA reads is an indicator that there's actually active transcription going on. With the idea that since there are diverse patient responses to a viral heart infection, there's likely clusters exist in the actual transcriptomes of these patients, which would represent adaptations to the virus or responses. So I performed a consensus clustering method and found that 3 stable clusters exist. And I compared each of these clusters to all of the virus negative hearts to get a host gene expression profile for these 3 different clusters. And so these clusters here are just represented in 2D space. Important to note that this is just a representation that I used a substantial amount more data to actually form the clusters. This is just for visualization. When I merged the data sets together, I batch corrected for the data set itself, as well as sex and condition. So dilated cardiomyopathy are healthy. So I was interested in general responses to the virus and not the covariates. And importantly, these covariates aren't enriched in any of my clusters, indicating the batch correction worked well. Interestingly, when I infer the ancestry from all of the hearts, that this isn't enriched in any of the clusters as well. And perhaps most striking is when I look at the virus type isn't enriched in any particular cluster, which indicates that these may be general responses to viral infection, regardless of the virus type. So I'm sure you're asking yourself now, what are the actual adaptations? What does the gene expression look like? The first one is the one that we would generally expect. It's inflammatory. There's a large enrichment in inflammatory processes. These are gene set enrichment pathways. And the second two clusters were a little bit more enigmatic. They are actually low for inflammatory processes. I did look in the literature a little bit and found there was a connection between inflammation and stress and AU-rich elements, which are mRNA motifs, the UAAUUU motif shown here, that exist in the 3' untranslated region of mRNAs. And so if an RNA has this motif, it can be bound by particular proteins and cause rapid degradation. So this is a form of post-transcriptional control. So that entire pools of RNAs can be either stabilized or depleted depending on the AU-rich element binding proteins. And when I look at the genes with the specific motifs, this strongly divides cluster 2 and cluster 3, where cluster 2, the upregulated genes are enriched for AU-rich elements, and cluster 3, the downregulated genes are enriched, which allows us to rename the clusters inflammatory, ARE up, and ARE down. This is a molecular definition for the clusters, but we wanted to figure out what was a more biologically relevant definition. So I did cybersort, which infers the cell types actually present in each patient sample based on gene expression patterns. We found that the cluster 2 was associated with increased cardiomyocytes and decreased fibroblasts and endothelial cells, which may represent mild cardiomyocyte hypertrophy because cardiomyocytes don't divide very often. And this has been noted to occur after some viral infections. We also noted that the inflammatory cluster was associated with an increase in fibroblasts and increase in immune cells, which kind of makes sense since it's an inflammatory cluster. And the takeaway here is that the ARE up shows signs of cardiomyocyte hypertrophy, and the inflammatory cluster may be having fibrosis and immune infiltration. I then wanted to look for classical biomarkers of heart failure. And when I looked for the typical serum biomarkers, I found that they were all enriched in the inflammatory cluster. Shown here, NPP-A and NPP-B are ANP and BNP, the classic clinical biomarkers for heart failure, which indicates that the inflammatory group may be on the path to heart failure. I then looked for matrix remodeling, which is also associated in a pathogenic response to viral heart infection, and found that a lot of mediators for matrix remodeling were also upregulated in the inflammatory group. So it kind of indicates that this may be one of the more negative responses to viral heart infection with matrix remodeling and heart failure. And so if we come back to the data science-based systems biology, we've taken large data sets and used bioinformatics to generate a testable hypothesis, and that is that hearts adapt to the presence of virus in one of 3 ways. So this is testable, and we can design experiments to actually test if this occurs. And so to do this, I wanted to create a simplified system that we can use a bioengineering approach. And so we took a homogeneous cell type, which were AC16-derived cardiomyocytes, single cardiopathogenic virus, which was Coxsackievirus B3, which is probably the most well-known cardiopathogenic virus. It infects cardiomyocytes directly and causes myocarditis. And the virus itself is an enterovirus, so it's positive sense RNA, non-enveloped virus. And importantly, I actually didn't find the Coxsackievirus B3 in any of the patient samples. So this will also be a test of generality. So if we can um, see similar trends here, it may reinforce the idea that these are not virus-specific responses by the heart to infection. The strategy itself is to create an in vitro model of chronic viral heart infection. So a previous graduate student in the lab who has since graduated took a maturation deficient version of Coxsackievirus and stably transfected the cardiomyocyte-like cells and isolated 12 clones, which will represent 12 separate infections. And since the virus is maturation deficient and only under the expression of a leaky promoter, you end up with low amounts of viral RNA, but no actual intact virions, which is reminiscent of an actual simmering heart infection. Can then do a differentiation protocol to make the cells more cardiomyocyte-like and do RNA-seq. And this allows us to test if the 3 patient-derived clusters are observed again, as well as experimentally manipulate if they are to gain mechanistic understanding. And so when I map the transcriptomes of these in vitro cell lines onto the transcriptomes of the patient samples, I find that it actually does map one-to-one, where there's an inflammatory group of the cell lines, an ARE up group of the cell lines, and an ARE down group of the cell lines. And that they actually, if you look at their differentially expressed genes, they also map one-to-one, ARE up, ARE down, and inflammatory. So we can conclude that all 3 patient sample adaptations reoccur in this in vitro model of viral heart infection. This just shows that the actual pathways that we used to originally define the clusters are the same in the cell lines, inflammatory, ARE up, and ARE down. But our real goal is to investigate the relationship between the adaptations and experimentally manipulate it. So it's important to define key pathways to manipulate, pathways that actually define the characteristic of each cluster. So we chose NF-kappa B as a defining pathway for the inflammatory cluster. And then we chose P38 MK2 signaling pathway for the defining characteristic, since P38 MK2 strongly regulates ARE genes. So we can ask several questions. Are NF-kappa B and P38 key regulators? Was our underlying structure correct? As well as will activating or inhibiting these regulators cause cluster switching? So this is a map of the NF-kappa B pathway, and we can use an I-kappa B alpha super repressor to inhibit the pathway, as well as use constitutively active IKK2 to activate the pathway. Similarly, this is the P38 pathway and how it relates to ARE proteins or ARE binding proteins. And we can use a pharmacological inhibitor of P38 or a constitutively active version of the upstream kinase. With the important note that high P38 activity should result in high levels of ARE mRNA, and low P38 should result in low levels of ARE mRNA. And so this is the general perturbation strategy. I took the CVB3 chronically infected clones, transduced with the constitutively active or dominant negative signaling gene, differentiate them, and do RNA-seq. Similarly, I can use the pharmacological inhibitor and do RNA-seq after differentiation. And so this is the second experiment, just the control cells. I have in the The colored dots are the centroid of the 3 clones that I picked in each cluster. And we can use this PCA to map actually what the perturbation is doing to the transcriptome of each of these clones. So if you can imagine, we start with the inflammatory cluster, and one particular perturbation could move it towards the ARE down group, it could move it towards the ARE up group, or it really could move it off into space. And we're going to find out. So when I activate NF-kappa B with the constitutively active IKK2, it actually moves off into space, and which is a little bit concerning at first. But when I looked at what this space represented and mapped it back to the actual patient transcriptomes, this was still within the inflammatory group. It was just an unexplored space in this the PCA plot from the clones, since they're have a lot lower variability between each clone. When I inhibit P30, or when I inhibit NF-kappa B, I find that in the inflammatory and ARE up groups both move towards the ARE down. And when I activate P38, actually nothing striking seemed to occur. However, inhibiting P38 similarly caused inflammatory and ARE up to move towards ARE down. So with this, I feel confident that we can conclude that P38 and NF-kappa B are key regulators behind these 3 adaptations or responses to viral infection, and perturbing them can cause indications of cluster switching, as well as ARE down seems to be a signaling ground state. So whenever you eliminate P38 or eliminate NF-kappa B signaling, everything shifts to ARE down. So if we come back to the wheel of systems biology, we've now done the statistical analysis and found that NF-kappa B and P38 drive the heart adaptations, and perturbing them can cause cluster switching, and that ARE down is a signaling ground state, which leads to a knowledge gap. What is the chronology of these adaptations? Do they occur sequentially? Do they occur at the same time? And so we generated and looked for some new data sets to address this question. Specifically, data sets that have a known onset of infection. So the patient transcriptomes that I showed previously have a completely unknown time of infection. And instead, we picked several data sets that have known times of infection, or relatively well known. And specifically, we picked acute CVB3 infection in cell culture, human hearts from people that died of SARS-CoV-2 infection, since SARS-CoV-2 is a known cardiopathogenic virus, and hamster hearts after influenza or SARS-CoV-2 infection, a full month after, where there's no signs of viral pathology remaining. So when we took a cardiomyocyte-derived cell line, the same one that we used for the chronic clones, and wildly overexpressed the receptor for Coxsackievirus, CAR, we infected these with CVB3, and we found that they actually strongly map to the ARE down group, which indicates that with a super physiological amount of viral receptor, you can end up with an ARE down adaptation. We then looked at the SARS-CoV-2 hearts from the autopsy and found that these strongly map to the inflammatory group. I found a couple other instances that map to the inflammatory group as well, such as mouse hearts with an acute adenovirus infection. And so short-term cardiopathogenic virus infection can cause a heart inflammatory adaptation. And the final data set we looked at was, as I mentioned, hamster hearts a full month after SARS-CoV-2 or influenza virus, which had no signs of pathology remaining. And these mapped to the ARE up group, which indicates to me that the ARE up may represent an actually successfully resolved infection. And so we can take all of the things that I've talked about together and summarize them into the key takeaways. We found that there are three heart adaptations and that are found in response to diverse viral infections and timescales, and that these are the inflammatory, ARI up, and ARI down, with the inflammatory being associated with matrix remodeling and fibroblast enrichment as well, and the ARI up being mild cardiomyocyte hypertrophy and resolved infection, where ARI down is a little bit more enigmatic and has a potentially dysregulated signaling state, since it's the ground state of all these three clusters. So if you remove any of the things that are propping up inflammatory or ARI up, it seems to shift to ARI down. We can also represent this as a Waddington landscape, where this dot is an uninfected heart. And if you have a viral heart infection, your initial response will be to upregulate NF-kappa B, putting you to inflammatory state. And then depending on the level of P38, when the NF-kappa B starts to subside, you'll either roll to the ARI up state or the ARI down state, with the potential for ARI up to shift to ARI down. But we really don't ever see ARI down moving back towards the other two states. And together, this is a framework for interpreting really how the host responds to viral heart infections. And with this, we've kind of taken a complete loop around the wheel of data science-based systems biology. And so this is a bit of a shift in the talk, and I'm going to talk about the other side of viral heart infection, so still very related. The first part was about how the heart responds, and now I want to shift gears and talk about actual like susceptibility to viral heart infection, so things that are there prior to the heart infection that may impact whether or not you have a severe infection. And to do this, I want to introduce a mathematical model of CVB3 infection that my lab published a couple of years ago. And it is an ODE-based model and encodes basically every piece of the viral life cycle from entry, replication to release, and includes viral antagonism by the intrinsic immune response. And one of the earlier things that I did when I joined this lab is I took this viral model and actually recoded it into a graphical user interface so that anyone who isn't familiar with MATLAB can still use the model and run experiments. And this on the right is a video of that graphical user interface showing kind of like how the model looks. And so when you run the model, you end up with a viral trace of virions over time. This is for 16 hours. You can turn off the ability of the virus to antagonize the host immune response and decrease the viral replication, which is the blue line. And you can also turn off the entire host antiviral response, which is the green line, among a wide variety of other things. You can change the polymerase rate, translation rate, and simulate antivirals. It's a really an interesting model, and I encourage you all to check it out if you find it would be valuable. And so with this CVB3 model, can we incorporate the patient diversity that I talked about in the first half of the talk? Specifically, the patient heart transcriptomics include host genes essential for viral infection. And the ones that come to mind are first are the CVB3 cell surface receptors, CX80R or CAR, and CD55 or DAF. But the problem is that these patient heart transcriptomics are RNA, and the model itself uses protein because it's a protein-based model. So we would need to infer the protein abundance from RNA to input in the model. And the strategy that we took was leveraging gene-specific RNA to protein relationships, which I'm going to talk about further. And so this project was really spearheaded by a graduate student in the lab, Andrew Sweat, who has since graduated, and it was really a pleasure to work with alongside him on this project. And so basically what he did is started with a mass spec TMT proteomics and used and so these were proteomics from 375 cancer cell lines, and he scaled them using two reference cell lines to more absolute quantification proteomics, label independent. So it was these were swath proteomics. And then with this relative amount of protein per sample, you can actually run a regression for the RNA per sample and generate models of the relationship between RNA and protein. And we found that there are three general relationships that RNA and protein have together, which is the median relationship, where the amount of protein really doesn't vary depending on the amount of RNA. And these are generally pretty long-lived proteins, or those in complexes. The majority of the proteins, however, fall under the hyperbolic to linear model, which is a trace shown here in magenta. And then some proteins are better modeled with the hyperbolic to linear model, including a lasso regression. So these are proteins that are better modeled when you include information about other genes in the sample. So other genes influence the level of the protein. And so with these three models, you can predict the abundance of a large number of proteins. We ended up with 4,366 proteins that can be predicted from the level of RNA. And included in them are CD55, which is the hyperbolic to linear plus lasso, and CAR, or CXADR, which is hyperbolic to linear relationship. And so plotted on here are the actual RNA-seq values from the patient samples. And you can see that they appear along the entire, or a large range of the possible protein concentrations. And so with this in mind, we can create individualized models. And so basically, we take each patient's CD55 and CXADR, or CAR and DAF, and input that as a starting condition for the Coxsackievirus model and run it out. And this is what is shown on the left, where by 8 hours post-infection, you don't see the production of a large number of virions, but however, by 24 hours, almost all of the simulated hearts are infected and producing virus with a mode around what the amount of virus we'd expect would actually lyse the cell. Interesting to note, about 15% of the hearts don't actually, or aren't actually successfully infected by 24-hour, implying that a substantial portion of the population may be intrinsically resistant to Coxsackievirus infection. When we look at the actual infected hearts, we find that they form three modes using the Gaussian mixture model with low, medium, and high susceptibility. And we can map these susceptibilities onto the abundance of CD55 and CXADR to see which of these receptors is really defining the susceptibility. We find that CD55 doesn't correlate a lot with susceptibility. However, CXADR strongly correlates with the susceptibility of a heart in these simulated infections. And we find that there is an inflection point at around 5,000 copies of CXADR is the kind of the minimum we see for successful infection. And this is actually a testable prediction. Is 5,000 copies of the Coxsackievirus receptor the threshold for susceptibility to infection? So we took an engineered cell line, the AC16 cells, that express CAR only when doxycycline is added. So we can add doxycycline for various amounts of time, produce a certain amount of the viral receptor, and then infect at that exact moment, let the infection go for a few hours, and see if it was see if it was successful. And so for this example shown here, we used 30 minutes of docs, which results in about 2,800 copies of CAR on the cell surface at the moment we add the virus. And when we add the virus, we actually don't see substantial affection beyond the parental. So it's a very an absolute minimal amount of infection. So these are not susceptible with 30 minutes of docs and 2,800 copies of the receptor. However, when we add the doxycycline for an hour, we end up with 4,800 copies of CAR on the cell surface, which is pretty close to the actual predicted susceptibility threshold. And we see that these are actually now substantially susceptible. They actually support viral infection. So this it is a validation that the model successfully predicted a threshold point for CVV3 susceptibility, and that there's a good evidence that the 15% of people below this threshold point in protein abundance may be actually resistant to Coxsackievirus infection. So with that, I want to say thank you, everyone, for listening, and I'll thank you to the Jane's Lab for helping out with this project, especially Andrew and Kevin.
Thanks, Cameron, for the nice presentation. I have just one about, you managed to infer some part of the dynamics of this infection via extrapolating by RNA-seq data, right? So you do some experiment
Mm-hmm.
to actually see how it happens to go down. What do you envision is a challenge to be able to be more comprehensive if you don't have an hypothesis, for example, of a specific receptor of a specific pathway that can affect your condition? How will you go about having some trying to extrapolate the dynamics in a broader context where other molecules, well, are sure involved, but how can we get rid of the noise, let's say, fundamentally?
Yeah, I think you're touching on an important tricky part, especially that these are human samples taken exactly one time. I think that probably the better way to actually figure this out would be through a clinical trial, which is obviously very tricky. On the plus side, have been some trials in various viruses that if someone has a viral heart infection in their biopsy, they can treat them with like interferon or something very generic, and actually restore some heart function. I think the real key with this work is I envision it where you would find biomarkers for these three responses to viral heart infection. And if you have the biomarker, you could potentially just shift the gene expression in the heart to a more productive response. And so basically, like, just take a very broad approach to it, since it appears as though the heart responds to this foreign genetic material in these three ways, regardless of the kind of virus, especially since it's a low simmering infection. I mean, imagine like a very acute infection, probably very somewhat different, but and that's basically how I see kind of working through the noise, is taking a very general approach.
I'm curious, the back and forth between the model and the wet lab, you know, where you see something in the model and you say, geez, you know, that we've never seen that in a wet lab. And then you go to the wet lab and you say, oh my God, how the hell did we miss it?
Yeah.
You know? The question is, are you aware of like, you have a shopping list of wet lab limitations? What are the things? I have a concern about over-investing in modeling to the cost of investing in wet lab research. And wet labs are very dependent on new devices, new technology, but also new reagents and stuff like that. And so are you able to identify, like, here's a list of things that we would like to look at. We understand it well enough from the model to know what we want to see, but we need somebody to fund the development of the wet lab so that we can validate in real biology what we're finding in the models. And
Yeah.
my bird and I will go silent.
I think that is a very good point. That it interests me a lot, and I think that there's kind of two ways to approach this, at least how we've approached this. There's one, you can do like a parameter sweep or a sensitivity, and see like which combinations of perturbations you haven't looked at before, and so basically like use the modeling to infer which data set you should generate. And I mean, obviously, the technological advances to create bigger and more trustworthy data sets to incorporate in the modeling is valuable. And the other thing that we particularly like is when we are building models, we like to find the point where the model falls apart, because that's usually the point that you don't understand enough and should do an experiment or, several experiments to figure out why the modeling just simply doesn't work at that point. And that's usually, so those are the kind of the two ways that I see them interfacing.
And quickly, I'll have to read your bio to find out, but are you a biologist who went computational or a mathematician that went biological?
I'm a biologist, so I have learned about bioinformatics and modeling during my postdoc.
I'll ask one, which is sort of a generic one. How specific is this workflow to the heart? I mean, you have at least persistent changes in gene expression in this in your tissues. If I were growing long epithelial cells that I inflected with IAV, would I expect to be able to follow essentially the same kind of workflow? Because I'm particularly interested in looking at this difference in genetic susceptibility, which you're identifying here. It's very rare to be able to actually tie genetic perturbations to these mechanistic models. And so that's particularly attractive to me to ask the question, how can you generalize?
Yeah, and I think that I'm actually really glad you asked the question, because my current one of my current big interests is rhinovirus. And so I have a clinical collaboration with rhinovirus here. And I find that you can actually do a similar approach using public data and find rhinovirus in a lot of samples, and then you infer responses to it. As well as rhinovirus is very similar to Coxsackievirus, so we can adapt the model of Coxsackievirus to rhinovirus, and kind of do a similar approach. I mean, it's obviously not the same because there's different dynamics and different considerations, but the similar ideas and the similar philosophy of approach definitely works, and that's where I'm very interested in pursuing in the immediate term.
Mine was a curiosity because I'm always looking at this again on getting some better insight on the sequencing or omics data. So you showed in the one of the first plot you did a PCA, right? So this is a linear composition of the abundance of some reads, or depending on what you do that. And that would be is perfect if you try to predict things that move linearly. Like, okay, I have a perturbation that goes up or down, more or less. That's, I know that is tricky, but it's just for discussion. How should we go about when we know or we don't know if some of the components are not really up or down? They might go up and down. I don't know. They have a non-linear dynamics whatsoever. Does the model will be robust anyway, or we lose some information? We can find ways to dig in.
Yeah, I think that you hit the nail on the head with one thing that I was very, very difficult in the analysis, trying to think of exactly how to represent it. We went with the linear one because it's most interpretable, but you can also do like just direct distances, a Poisson distance between every gene in your samples, and so that can take into account these up and down dynamics. But I think that yeah, it's an interesting field, and I'd love to learn more about how, you know, what truly is similar, or how is the perturbation affecting it on a global scale as opposed to just the spot that we're looking. But yeah, I think it's definitely a good point.
I guess we all get there because we all are in the same field in this case. It's always tricky, but it's very interesting, I think. Yeah. Thank you very much.
I mean, I think John alluded to this in a sense. Perhaps it's not surprising that you get an acute inflammatory response and then you get some kind of post-inflammatory state and then a recovered state. I mean, it's not obvious a priori that it's going to happen that way, but a posteriori, it's not too surprising.
Yeah.
Are there other situations in which you might either within your and you found essentially two discriminators, right? You had your NS-kappa B and your P38. Did you try to break it down to look for subgroups within your three clusters? Because there was a certain amount of overlap, right? The
Yeah.
clusters were not very well separated. They made
Yeah.
sense, but they weren't very well separated. Could you identify substates, or was that not something, at least in vitro, maybe that your population is too uniform? I don't know if were there any other discriminators that came out at all that would.
Yeah. No, I think, and you raise a very good point. There were also indications that there's like potentially like a larger number of clusters, like 7. They tended to be very similar. Like it basically, like you would have multiple clusters with a unifying theme. So that's why I was confident that the 3 were probably the most straightforward way to describe it. But there were certain things like only a subset of samples had, like for instance, B cell related genes, like actual like IgG or IgM expression. And so those could be potential limiting factors as well. And I think this really touches on a big challenge of this sort of research is, if you have all of these tools like gene set enrichment analysis, how do you decide what path is most important to go down to differentiate a sample group from another sample group? And so I have my favorite methods, but this is something that I always grapple with, because like you could go down and, you know, someone could analyze this data and pick a different one and describe a different feature of these samples, which may also be important. And so I think that's, you know, I guess the joy of research.
Ellen, go ahead.
Yeah, hi, great talk. I have a question about the data sets you're actually using from public repositories, because I noticed, I took a quick look at your bioRxiv paper and you mentioned using some GTEx consortia sequences. Are you actually having to, because with human data, they don't typically publish the FASTQ files because of the de-identification of genomic or any kind of sequence related data. So are you having to actually request the original data in order to be able to find viral sequences?
Yeah, so there was 3 data sets. One of them was completely publicly available. GTEC did require a request, and then there was a European data set which required a very difficult request. Um, but yeah, basically to do this kind of approach, you have to have the raw FASTQ files.
Yeah. Okay.
Yeah.
That's what I just wanted to double check, because I was like, how did he do that? That's really cool. And we've thought about doing some of those kinds of things too, but it's like, as far as I was aware, you had to be able to request the original files.
Yeah.
the de-identification issue. So yeah.
And that is also an important difficulty, is that a lot of the questions that we had been trying to publish this were like, what are the actual clinical phenotypes? And that's way more difficult when you just are having public data, and so you have to like go back to the authors and ask if there's more data available, which can be a big challenge.
Yeah, I've been part of the Human Biomolecular Atlas program, and one of the things that we had to deal with is how much donor metadata could we actually put up with even the processed sequences and things, because otherwise too much and you get back into that realm of, oh, we could potentially identify this person, right? So yeah, so but yet at the same time, it seems like there's so much information that could be used. Like, I love what you did with this. This is so fantastic. I don't know if you're aware of this, but cats actually have cardiomyopathy or that dilation happening a bit. And at least the vets that I'm aware of were not aware of the root cause. And I'm wondering now that I see this, if there isn't a viral component with that as well, that happens to be pretty awful in cats, actually. So something to look at.
Yeah, that's interesting. And my gut reaction is I would be curious to see if it was also a coronavirus, because the coronavirus research in cats has been going on for decades.
Yeah.
It's a huge problem in cats.
Yeah. You might want to see what you can find with heart, if there's any heart-related data sets for cats, because I know this seems to be a big problem, and I don't think they've put like this together. Like, you've put this together, and this is fantastic and really interesting, and something I hadn't seen before, you know, like putting the viral component in the cardiomyopathy together, and it's just like, oh man, that's insane, you know. And also that even in people who appear normal, that they can still be having this effect, you know. So that's actually also super interesting.
Well, I wonder, I mean, the veterinary data sets, if you could get access to them, you won't have the de-identifications, the serious de-identification issues. And so you might actually be able to do that correlation with clinical outcome more effectively in an animal model.
Yeah, no, that would be interesting. Yeah, I'll have to look that up. I had no idea that cats got cardiomyopathy.
Oh, really badly. In fact, I think one of my own pets may have, I mean, definitely died from this, and it's pretty, like, they can seem fine and then just literally keel over. So it's really awful. It's kind of like a silent killer, but happens fairly frequently in cats, apparently. So
interesting.
Is the genetic variability in cats as broad as in humans? I know the American Shorthair is a fairly inbred.
Yeah. No idea.
Yeah,
I wonder if the susceptibility but that would be a great one.
Yeah.
I don't know what the availability of feline data sets is. It's always a tricky.
Yeah, I'm not sure either.
No, I'm not, but after seeing your talk, this is something that I would definitely be looking for now. It's like, oh, that's really cool. So
just a comment that on the idea of studying certain pathologies that are more prevalent in certain pets, we can actually discover something about human disease, and there might be natural model system where we don't have to experiment. It happens, and it's relevant in cancer. We are exploring this also in cancer. There are certain cancers that are more prevalent in feline than in other, or in dogs. So that's very exciting. There is this idea of One Health, and some funders are also looking at that.
I mean, of course, there's also the direct concern for animal health, especially for companion species. It's a big topic, because I think people realize that veterinary medicine is not always as developed as it should be. May well be, as Ellen says, a real missed opportunity among the veterinary scientists. That's really exciting. Okay, well, I want to thank you. I think we had a great conversation. It was fascinating work. I'm looking forward to seeing that publication when it comes out. I saw
Hopefully
it.
very soon.
That's great. And maybe you can come back in a bit and tell us what you found in going forward and in the context of the respiratory context. Okay.
Love to.
Well,
Thank you for the invite. It was a really good conversation.
Okay. Thank you, everybody. See you again in 2 weeks.